So hi friends, so this is just a continuation part of the part 2 video which I posted before. So what we are going to see now is we are going to see the three different types of pandemic coronavirus which was outbreak and why it has been in the state and what kind of issues we are facing because of all those things. That's what we are going to see in this video. So if you take SARS, the full form of SARS is severe acute respiratory syndrome. So this pandemic outbreak was happened in 2003, and it has been believed that the virus was the origin form of the virus is from the bat, and it has been passed on to one organism. From that it has been passed on to the human. So that is what they believed for. And similar way, the MERS means Middle East Respiratory Syndrome. Here also the same kind of origin they have found. They have found out that it has been originated from the bat, and from the bat it has been passed on to the humans. If you take the COVID-19, the current state, current issue which we are facing now, the similar thing, from the bat only it has been passed to the humans. So this is all the belief of the scientists or the researchers, and there are a lot of proofs regarding to this. So here what is the role of the bat over here? So the bat acts as a natural reservoir. So here what happens is whatever the virus, especially the coronavirus, it actually stays in this place and it tries to mutate, or all the replication takes place inside the bat. Okay, once there is a mutation that happens, and that which can adapt to the another organism means, then this virus will hop over to another organism. So that what has taken place, there was a mutation which happened in the virus which is there in the bat, and that made the virus to hop on and move to the civet. And from the civet there was a mutation which happened in the virus, and that made the virus to hop over to a human being. Okay, so this is what the case for the SARS. Same thing in the MERS. What happened? The same mutation happened in the bats, and from there the virus has hopped into a camel, and in the camel there was a mutation, and it has hopped from the camel to the human beings. Whereas in the case of COVID-19, it has been believed that it has been passed from the bats to the pancreon, but still the research is going on, so it is not confirmed. So from the pancreon, it is believed that the mutation has happened, and from that it has been passed on to the human beings. So this is what the research data shows. Now we'll see what is COVID-19 and where it has been actually originated. So this is all the kind of information which I got from the net and different websites. So it is shown that especially COVID-19 is being believed that it has been spread from the Wuhan city of China. So everyone would have known that. So that is what the belief. And the next thing we need to know about the COVID-19 is it actually causes the severe acute respiratory syndrome. So this is what the main problem which the COVID-19 causes. So before moving forward, before knowing the different pathophysiology or about the unique feature of the COVID-19, we need to know the main difference about the DNA virus and the viral DNA. Uh, RNA virus. Okay, so what is the major difference? Why there are a lot of importance is being given to a RNA virus? If you see here means, if you see a DNA virus, it is mostly double stranded, and here it will be mostly single stranded. In rare cases you can see double stranded RNA. And if you see here in the DNA virus, if you see here the genome size will be big, and it makes the virus, the replication of the virus over here is much slower when you compare with the RNA virus. So this is what all the major difference. So what happens? So if the genome size is lesser, the replication power is speeded up over here. So that is also one of the problem. The next problem is the DNA virus have a proofreading. So whatever the replication is being done, it will be proofreading. So there will be not much of mutation which happens in DNA virus. If you take an RNA virus, there is no proofreading. So whatever the replication which happens will be not proofreaded, so there might be any some errors in the replication process and it results in the mutation. Okay, so that is what the main problem what we are facing now. So if there is any mutation which happens in the RNA virus, what happens, whatever the vaccine or whatever the medication which we are able to find out, it might be different for the new strains of the COVID-19. Okay, so that is what the main issue of an RNA virus. So major thing is RNA virus has a speed enough power of replication and there is no proofreading, so there will be a lot of mutation when compared to the DNA virus. Okay, so these are the two main important points which you need to remember before we are going to discuss about the COVID-19's unique feature. Now we'll see the unique feature of the COVID-19. So if you compare with the other type of RNA virus, in the COVID-19 you have lot more of genes, more genes are there. So that makes the COVID-19 strong. Okay, so what happens over here? So because of this more genes, there will be lot of mutation. So there will be lot of harmful mutation which results for us. So it might be good for them, but sometimes it will be bad for us. So this is what happens in a COVID-19. But there's a specific enzyme which is present in the COVID-19. What does this enzyme do? It tries to go and check, okay, if there is any replication error or something, it will go and check and it tries to correct the replication error, maybe correct the mutation which has happened. If there is anything wrong, it try to stabilize it. So what happens if it's able to, this enzyme helps to stabilize the COVID-19 when it is in a good sign for us, right? Because there will be not of there will be less of mutation which you can see. So that is actually a good sign with regarding for the development of the vaccine or maybe for the development of the new medicines. So what, not only that, for the development of the medicine, it also help our human body, the immune system in our human cell to recognize the similar kind of virus and try to attack them. Okay, so this is what a good sign. So what is the unique feature of the COVID-19? It has more genes, so it might result in a lot of harmful mutation, but there's a specific enzyme which is present, try to check any replication errors and try to repair or correct the mutation. So in that state, it try to stabilize the COVID-19 state. Okay, that is actually a good stage in regarding with the development of the immune system or in the development of the vaccine. So now we'll see how this COVID-19 is being spread. So that's what we are going to see. So mostly the COVID-19 is spread by the air droplets and X1 is through the fecal matter. If there is any sewage leakage or something, if the person who was infected with the COVID-19 and that kind of feces is being released to your place, then that is also a source where you get this COVID-19 infection. Okay, and why the government or the health professionals insist you to wear face masks or insist you to wash your hand is mainly because of these reasons. So what are they? So if you take the air droplets, if when a person who's infected with the COVID-19 sneeze or cough, the air droplets, whatever it is there, whatever you release, will stay in the atmosphere for almost 3 hours. So if the another person, healthy person who tries to go in the same place, okay, then this person also might get the contact of the COVID-19. So that is main reason why the government is insisting you to wear the face mask, or face mask also not only enough, you need to wear the protective shield and all if you are passing into a very risk oriented places. And next, if you see the latex and aluminum sheets or aluminum plates, it almost remains there for almost 8 hours. Similar way, if you take cardboard, it remains there for 24 hours. And if you take counterparts or stainless steel or plastics and or if you see, it almost remains there for 3 days. And if you compare wood and glass, it almost remains there for 5 days. So that is all the main reason why the government insists you to wash your hands with soap or maybe the sanitizers. Okay, so this is how the COVID-19 is being spread on from one person to the other person. And if you see the more percentage of the mortality for the up to this June 7, it's showing like 5.74 percentage of the mortality is showing. But it might reduce the mortality percentage because still now we are not able to find out the exact number of cases which is there in the worldwide. So might be that is the reason why this high percentage of the mortality you can see. But if the number of the cases arises and the number of the death is reduced, then the mortality percentage also can be reduced. Okay, so now we'll be saying what about the R naught and the serial intervals of the COVID-19. So if you see, R naught means it's nothing but it's a rate of the speed how this COVID-19 is spread out into the population. So if you take a flu virus or influenza virus, here the R naught you'll see as 1. That means a patient who is infected will be able to infect another one person. So if you see here in the white picture, you can see one person has a power to infect the other person, and that person will be able to affect the another person. So if you take 3 generation, almost only 4 people will be affected in the flu virus. Okay, whereas if you take the COVID patient or the COVID state, here what is the status? R naught is almost around 2 to 2.5. So that means the patient who is suffering from this COVID-19 has the capacity to spread to 2 or more than 2 patients. Okay, 2 persons. So here if you take 3 generation, what has happened? One person has spread into 3, and this 3 person has a capacity to spread to the 9 persons, and from the 9 person to the 27 persons. So altogether if you see, it will be more than 30 or 35. So this is for the number of the persons. If you take in a long run, the number of the person who is affected within the 14 or 24 days is more. Okay, so this is for the severity of the COVID-19. So, and to be believed, if it's R naught is below 1, then it is actually, it's not a pandemic. It can be easily treatable and you can actually reduce the type of virus. If it's R naught 1, then whatever the case which is there, it will be in the stationary phase. It will be stabilized one. If it's above 1, if R naught is above 1, then that is a stage where we need to give lot of importance, and proper isolation or quarantine or social distancing need to be maintained, so that you can try to reduce the growth of the curve. Okay, so I hope you are clear with this R naught and the serial interval. And one more thing, the incubation period for the COVID-19 is almost 14 days, but for some persons it can be asymptomatic also. The person will be not having any symptoms. Only after 14 days or so they might get some symptoms, because they might be, the immune system of their body might be strong. So that is also one of the reason. Okay, so the next what we are going to see is about the pathophysiology of the COVID-19. So what happens when this COVID-19 pass from the upper respiratory system to the lower respiratory system? There are two types of cell which is present in the lungs. One is the pneumocytes type 1 and the second one is pneumocytes type 2 cells. So what is the function of the type 1 and type 2? Type 1 cell actually helps in the gas exchange. Okay, it tries to help you to exchange the oxygen and the carbon dioxide in and out. And what is the function of the type 2 cell is? Type 2 cell function is a surfactant. That means it helps to maintain the surface tension in the lungs. So that is what the function of the type 2 cell. Okay, so what happens is when the virus is invaded or when the virus enters the lungs, this virus mostly goes and attached to the pneumocytes type 2 cells. So what happens when this virus goes and attached to the cell? So that is what we are going to see now. Okay, so assume this as a cell. Okay, so what happens is this virus goes and bind to the specific receptor in the pneumocytes type 2 cells. Okay, so this is the receptor, and the receptor is angiotensin. Okay, this virus goes and try to bind to the specific. So what happens? The virus goes and bind to the specific receptor, and through this receptor, the single-stranded RNA, okay, with the positive sense, enters into the cell. So what happens once it enters to the cell, it goes inside the ribosomes and convert it into an mRNA. Okay, so that's what it takes place. Once the single-stranded RNA is converted to mRNA, this mRNA with a process called as translation transfer to the form proteins. Okay, so these proteins will be able to produce all of different types of polyproteins. Okay, so what happens once the virus tries to attach the specific receptor, when it attaches to the specific receptors, what happens? The single stranded RNA is released into the cell. Once it is released, it's enclaved into the ribosomes and tries to convert it to mRNA. Once it's converted into mRNA with the process of translation, it tries to convert mRNA to protein. So it can produce different types of polyproteins. And this polyproteins with the help of enzyme, okay, called as proteinase, tries to produce the different components which is available for the virus. So what are the different components which is there? One is the spikes. Main thing is the spikes which is present. So it tries to produce as different types of spikes. So there are two different types of spike. One is blue and a pink one. So as spikes. So different types of spikes, protein spikes it produces. The next it produces various enzymes, then the capsid. Okay, so this will be produced. Over into the next thing, this mRNA. What is mRNA? mRNA will be converted into the different or numerous amount of the single stranded RNA with the help of a different enzyme called as RNA dependent RNA polymerase. So with the help of this enzyme, the mRNA converts into the multiple copies of the single stranded RNA with positive sense. Fine. So this is what the main enzyme which plays a major role in producing or maybe producing numerous amount of the virus. So once this single stranded RNA and the different components of the virus is combined, it produces the numerous amount of the virus and it will be released to the different parts of the cells in the lungs. Got it? So this is what happens inside a pneumocyte cell. Now what we are going to see, okay, this is all the virus when it enters. What are the role of the virus or what are the things which the virus does to the body? What does our body does against this virus? That's what we are going to see now. So here what you can see is once the virus is invaded, is released into lungs, what happens? There are immune system which is there in our human system. So what it does is it tries to trigger the toxins or triggers the immune cells to the particular place. So here the macrophages, okay, what happens? This virus triggers the macrophages to produce a different leukins, interleukins. So interleukins 2, interleukin 6 and TNF alpha. This will be produced. Once this is produced, what happens? This will be released into a bloodstream. When this kind of cells is released to the bloodstream, what happens? It results in the vasodilation. That means it's expansion of the particular area. Then what happens? Increase of the capillary permeability. That means it helps in the easy passage of the, maybe there will be there will be easy passage of the fluid passing from one layer to the another. So this is results. So what happens if there is results in vasodilation, increase in the capillary permeability, then it results in the increase of the edema in the alveolar region. Okay, so this is what happens. Once this happens, what happens? The surfactants which is present in the lungs, that will be reduced. Once the surfactants is reduced, what happens? It increases the surface tension in the lungs. Once the surface tension is increased in the lungs, what happens? It makes the lungs to collapse or there will be a shrinkage of the lungs. Once the shrinkage of the lungs is there or there may be in the alveolar region, what happens is it results in the shortness of the breath or the speed of the but respiration will increase. So this is what results in. It results in hypoxemia or dyspnea. So this is what the condition, or this is one of the symptoms where the COVID-19 you can see. Okay, so this is what the macrophages does. What is the next thing which the other cell which does? So next what happens is this interleukins TNF alpha triggers the neutrophils, and it makes the neutrophils to enter into the lungs. From the bloodstream it makes to enter into the infected sites. So what it tries to do, the neutrophils try to enter to the targeted site where the infection is happening, and it targets the virus and try to kill the virus. So along with targeting this virus, lot of other pneumocytes type 2 cells and type 1 cells also get damages. When the pneumocyte cells is damages, what happens? There will be a shortening, or there will be difficulty in the gas exchange. Okay, once if there's a lot of built-up of the immune cells in one area, what happens? There will be inflammation or consolidation of all molecules in that particular area. That results in the one of the other symptoms called as cough. You might be coughing because of the difficulty in the breathing. There are a lot of molecules, blockage is there. So because of that, you will cough. So cough is also one of the symptoms. This also results in shortness of the breath, or maybe also increase of the respiration rate. Okay, and next what happens? So the same thing, IL-2, IL-6 and TNF alpha, what it also does, it tries to give an indication to the hypothalamus, which is in the central nervous system. The hypothalamus is something which actually tries to maintain the body temperature. Once it triggers the hypothalamus, what it tries, it makes the body temperature to rise. Once the body temperature rises, what happens? It is an indicator for showing a fever. Okay, so fever is also one of the symptoms of a COVID-19 patient. Fine, you are clear with it. Now we are going to see what happens. So this is all the conditions. Okay, so if there is a reduction of the oxygen, low of oxygen is there, means it triggers the chemoreceptors. It tries to activate the chemoreceptors. This chemoreceptor triggers the nerve system, and this nerve system, what happens, it tries to increases the heart rate and the respiration. So this is also another symptom related with the COVID-19. So what are the different symptoms which will be reaching? Will be getting fever, maybe cough because of this consolidation of all molecules, and maybe because of the increase of the edema you will be getting the shortness of breath, or maybe the respiration rate also will increase. And because of the chemoreceptors, it tries to increase the heart rate also. So these are all the various symptoms which you can see for the COVID-19 patients. Okay, so once if you reaches all this stage, then what happens is you might be in a septic shock, or that might result in the multi-organ failure. So once if you reach this kind of stage, it is very difficult to treat, and the percentage of the death will be more higher over in this stage. So this is one of the serious condition what the COVID-19 patients are facing, and this might result in putting the patient in ICU, ventilator. You need to get a proper kind of treatment in this kind of scenario. Okay, now we are going to see what are the different diagnoses which is available for this COVID-19. So mostly first they try to take a swab from the nasal region and from the oral region, and they try to cultivate it, and they try to see other if there is any other virus which is causing this common cold symptoms. They try to rule out that type of virus. Once this has been ruled out, then they'll try to do an RT-PCR, reverse transcriptase PCR. The sensitivity over here is only 70%. So if there is any doubt over there, they try advise you to take the CT scan of the lungs. Okay, so CT scan of the lungs, it shows a clear picture where the virus infection is there and what are the conditions, what is the current status of your lungs also is visible in this stage. Or the next one is the isothermal amplification. This is a rapid test. You'll be able to get the information within minutes. So this is also one of the tests which is being used. Next is the serological rapid serological testing. Here what they do is they try to find out the antibodies, but in this, one of the other positive thing is, if you have been affected before also, it shows us the antibodies which is present. So this way you might come to know, okay, the antibodies are present, or maybe if you have come across a similar kind of infection in the past days. Okay, so this is what the different diagnosis which is available in the current state. Next we are going to see is the what are the different strains of the COVID-19 which is present. There are almost 11 strains of COVID-19 is there in the current state, and the type O strain was the ancestor one which was from the Wuhan city. And now in the current world where the strain has been rotating is the type A2A is the current strain which has been causing this COVID-19. And there is another strain which is A3 strain. This strain is mostly seen in the patient who has a travel history from the Iran. So got what are the different strains of the COVID-19 also. So what are the treatment which is being given for the severe cases of the COVID-19? So remdesivir. Okay, this is actually an antiviral drug which treats for the Ebola. This was actually used. So this kind of drug is being used for the severe symptoms or severe cases of the COVID-19 patients. And for them, this kind of drug has been used. And next is lopinavir and ritonavir. This is also another antiviral agent which is being used in this treatment of the COVID-19. And next one is the chloroquine and hydroxychloroquine. This is actually an antiviral drug mostly used for the malaria thing. This has shown a promising result in the for a trial. So that is what the reason why it has been used for the COVID-19 patients. And next, there is another combination also with the hydroxychloroquine plus azithromycin. So this is also being used for the treatment of the COVID-19. So the FDA for the current state, this has been given approval, but it is not a proper approval for the current state because of there is no medication. This medication is being used for the severe cases of the COVID-19. So thank you for listening. So if you have any doubts related to this, try to post your doubts in the comments. I will definitely try to reply all to your doubts. So do subscribe to my channel so you'll be able to get further videos related to the science background. Thank you.